Hello students, welcome to EPG Pathshala. I am Vidya Kothekar and a former professor of biophysics from All India Institute of Medical Sciences, Delhi. I had been professor of bioinformatics at JP Institute of Information Technology at Noida. I had also been professor of bioinformatics and also director of Dr. D.Y. Patil Biotechnology and Bioinformatics Institute. But what is my real identity is a teacher, and I like teaching. Now today we are going to discuss about a module, DNA protein interactions, under the paper biomolecules and their interaction. Variety of proteins like polymerases, transcription factors, nucleases, histones, and so on associated with DNA. Some of these molecules recognize base sequence on DNA, while others, like histones, do not recognize. Attachment of endonucleases at a specific site, repressor-oppressor interaction are the few examples of a recognition of a DNA by the proteins. What are we going to learn in this module? Many things. We first start with the basic concepts in DNA-protein interactions. Then we like to see non-selective binding of proteins with DNA. Later, I would like to explain you the recognition of DNA base sequence by specific motifs like Helix-turn-Helix motif. Then we'll see recognition of DNA base sequence by zinc fingers. I want to also explain you interaction of Leucine Zipper, BZIP and BHLH with DNA. We want to see the binding of proteins to single-stranded DNA and recognition. That is very, very important about the viral interactions or interaction of viruses with other molecules. One of the most beautiful works on protein DNA interaction is the interaction between 434 Cro and 434 Repressor with their operator. Stephen Harrison at Harvard University showed that recognition of different operator regions depends more on other factors than amino acid sequence in recognition allele. This was most important because it was contrary to our normal assumption that it is the protein side chain which interacts with the specific residues in the DNA molecule, and their observation was just contrary to it. The 314 base pair regions that 434 repressor recognizes on the right hand side of the operator are not perfectly palindromic. Those on the left hand side are close to it. The synthetic DNA sequence used by these authors was closest to the sequence of OL2 with only one difference. There was one inversion where AT was replaced by TA. Basic concepts in DNA-protein interaction. DNA base pairs GC, AT, CG and TA have characteristics electron donor acceptor pattern in major and minor grooves. We've seen it earlier. The DNA binding proteins have DNA binding domains and special affinity to a single or double-stranded DNA. DNA backbone and protein interaction. The sugar phosphate backbone can participate by electrostatic and stacking interactions with proteins and lead to structural modifications in DNA. There are a number of direct and or mediated through water and ion contacts. The positively charged side chains of arginine, lysine, histidine can H-bond with electron acceptors, whereas negatively charged side chains as ASP, GLU can interact with H-bonding donors in the grooves. Protein side chains base interaction. The polar neutral amino acids such as serine, threonine, ASN and GLN can have more than one hydrogen bond depending on their orientation. Cysteine, selenocysteine, glycine and proline can offer recognition through spatial motifs as zinc finger. Hydrophobic side chains, short and long amino acids, alanine, valine, leucine, isoleucine, MET can participate in hydrophobic interactions. Aromatic residues, tyrosine, tryptophan and phenylalanine can have stacking interaction with DNA bases. Protein motifs involved in DNA interaction. Size-wise, alpha helix is the most suitable motif for interaction in the major groove of DNA. Antiparallel H-bonded beta sheet can fit in minor groove with one strand recognizing DNA base sequence similar to non-intercalating drugs. For double-stranded DNA, aromatic amino acids, phenylalanine, tyrosine, tryptophan exhibit specificity. There is only partial insertion of these residues, which causes bending of double-stranded DNA. Latter decreases Tm because of energetically favored kinking. A very interesting analysis was done by Luscombe based on the structures of DNA protein complexes. They analyzed 129 structures. The three-dimensional data was analyzed and tried to find out what interactions between protein and DNA in this 129 protein DNA complexes were present. The summary is presented in the figure on the right hand side. Interestingly, 2/3 of interactions were due to van der Waals contact, no specificity. 1/3 each were due to H bonds and water mediated contacts. Latter also were usually non-specific and acting as space fillers at the DNA protein surface. You can see the interaction, the majority of them between arginine and guanine. Then there are interactions between the lysine, guanine and adenine, interactions between the base pairs, that is arginine interacts with 80 base pair, 80 base pair, 80 base pair, and so on. Many proteins have non-selective bindings with DNA. We have already gone through the example of histone DNA interaction. The picture is shown in the right hand side of the DNA binding to histone proteins. In nucleosome, there are over 120 contacts between protein and DNA, and they are unevenly spread between helices alpha 1 and alpha 2 and L1 and L2 loops. There are many salt links, hydrogen bonding, and water-mediated interactions. These are important, but all of them are non-specific. Recognition of these DNA-based sequences by Helix-turn-Helix motifs. Many of the DNA-binding proteins have Helix-turn-Helix motifs. The proteins that regulate transcription of DNA recognize DNA-based sequence through discrete binding domains of less than 100 amino acids. Many prokaryotic regulatory proteins contain Helix-turn-Helix motifs that recognize and bind to specific region of operator of DNA. One of the classic examples is of Cro protein. Repressor and Cro protein operate a prokaryotic genetic switch region. 3 related species of temperate bacteriophages, lambda, 434 and P22 have been studied. We show in this picture the genetic switch in Cro repressor. There are 3 regions, OR1, OR2 and OR3. The repressor binds to OR1 and OR2. It turns off the synthesis of Cro protein because repressor acts for its own synthesis by facilitating the binding of RNA polymerase, shown in the lower slide, to its own gene. When Cro binds to OR3, it blocks binding of RNA polymerase to repressor promoter. The transcription of phage gene to the right can only occur. Now let us see the details of this recognition. We show in this picture the sequences of OR1, OR2 and OR3. The nucleotide sequence in this 3 region is palindromic and quite similar but not identical. The 2 halls of binding site are related approximately by 2-fold symmetry axis, shown here in the red color. Both Cro and repressor are dimers. The palindromic part of OR1, OR2 and OR3 provides almost identical recognition sites. Structure of Cro protein. Matthews et al. Determined structure of Cro protein from bacteriophage lambda at 2.8 angstrom resolution. Each unit is of 66 amino acids. It folds into 3 alpha helices and 3 antiparallel beta sheets. The protein belongs to the class of alpha plus beta family. We show here the beta sheet region from the two subunits, beta 1, beta 2, beta 3 and beta 1, beta 2, beta 3. The alpha helices 2 and 3, residues 15 to 36, form a unique helix-turn-helix structure. Dimerization of the protein depends primarily on beta strand 3 from both monomers. We saw that in the previous slide. These strands at the carboxylic end are aligned in anti-parallel fashion. It was very difficult to co-crystallize proteins with DNA in the initial stages. So, model building of Cro DNA interaction was attempted. Brian Matthews showed how the two recognition helices of Cro dimer can be fitted in major groove of DNA. Amino acids from alpha 3 helix can then make contacts with the edge of base pairs in major groove. Binding of lambda Cro with DNA. A schematic space filling model of dimer of Cro bound to bend DNA was built by Ollendorf, showing the interaction of two alpha helices in the major groove of DNA, which is shown in the picture on the left hand side. The recognition helix makes contact with half of the palindromic sequence. The second helix in this model binds to second palindromic sequence, and the recognition of Cro protein was because of Helix-turn-Helix motif. Specific amino acids in recognition helices were important because they made specific contacts with nucleic acid bases. Subunit interaction that provides correct distance and relative orientation was also found to be very important. The model was in agreement with genetic study. Binding of Helix-turn-Helix motif of lambda repressor to DNA. The structure of operating binding domain of lambda repressor was determined by Pabo and Lewis in 1981. Bremer and Pabo in 1991 obtained the structure with cognate DNA sequence, which is shown in the picture here. The PDB ID of this structure is 1LMB. The Helix-turn-Helix and the loop region conformation was very much similar to Cro. The two recognition helices could fit in two adjacent major grooves of DNA as proposed earlier by by Pabo and Lewis and Ollendorf's model seen in the previous slide. Zinc fingers. The classic zinc finger was described in the laboratory of Aaron Klug at MRC UK from amino acid sequence of transcription factor TF3A from Xenopus levis that controls transcription of ribosomal 5S RNA. The 344 amino acid sequence of TF3A contains 9 repeats, each of 30 residue each. The sequence is characterized by 2 cysteine at amino end and 2 histidine at carboxylic end and an intrinsic zinc ion along with it. Nucleotide sequence in Bacteriophage 434 in OR1 OR2, OR3 and OL1, OL2, OL3 is shown in this figure. Both Cro and Repressor are monomeric in solution but form dimers when these are bound to DNA. The proteins impose precise distortion in DNA structure. DNA is in B form and overwound in the center and underwound at the end. The distortion narrows minor groove at the center and widens major groove at the end. There are contacts between amino acid side chains of recognizing helix and DNA base pairs. We show in this picture, in the left picture, left of this is a normal B form of DNA, and on the right of that is a DNA structure after binding of 434 repressor, and you find narrowing of the minor groove and widening of the major groove in this picture. The right picture shows how the recognizing helices shown in green and blue bind in the major groove. Recognition of DNA base by 434 Cro and 434 repressor. There are several contacts between protein side chains and DNA bases. Although GLN29 and GLN28 are important for binding, they do not give specificity as the first two base pairs in all sequences were identical. So you cannot call that these are specific contacts. However, the methyl group of threonine 27 and GLN29 form being a hydrophobic pocket to receive the methyl group of T12 is responsible for recognition. Let's see more about the recognition of DNA by 434 Cro and repressor. DNA distortions created by both these proteins were not identical in different complexes but similar in both the halls. Binding of repressor and Cro induces different shapes of distortion in the binding region. In case of repressor, two distortions induced by proteins are similar in OR1, while in case of Cro, these are similar to each other but different than repressor. In case of binding of repressor to OR3, the two distortions are different and hence repressor cannot bind as tightly, while Cro binding to OR3 is stronger. The mechanism of recognition of 434 Cro and repressor is explained in this figure. Like we show the model on the left hand side and on the right are shown the distortions. Like when in the figure A, it is the binding to OR1 by 434 repressor. The square part shows the distortions created by them. B is the binding of 434 Cro. It creates distortions which are similar in both halls but different than in the case of repressor. In figure C is OR3 binding to 434R. Now the distortions have different shapes and hence the binding of 434R is weaker. Recognition of DNA by eukaryotic transcription factor. The transcription regulation in eukaryotes is more complex. The process is controlled by complex set of regulatory elements called transcription factors. We shall focus our attention on specific transcription factors as zinc fingers, leucine zippers, basic region leucine zippers B-zip, and basic region Helix-Loop-helix BHLH motif. Single-stranded DNA binding proteins. As you know, there are number of proteins that bind and recognize single-stranded DNA, and these interactions are very, very important. These are identified both in viruses and in organisms from bacteria to human. A typical example is of simplex virus HSB1. The single-stranded or SS DNA binding protein, the head of 8 helices, binds and recognizes herpes simplex virus. The back of this protein has a very typical structure. It has 3 beta strands. The shoulder contains a helical and a beta sheet region. These interactions prevent a premature annealing between the 2 strands, and that's why that interaction is very important. Classic zinc finger. The classic zinc finger is a motif that is repeated in tandem to recognize DNA sequence of different lengths, with each finger interacting with a specific base sequence. Other zinc fingers bind as a monomers to discrete sites or as dimers to palindromic site. The strength in binding can vary depending on the sequence of both protein and DNA and length of spacer between the fingers. This gives a very high level of recognition. We show in this picture a zinc finger with 2 cysteines and 2 histidine. It's a classic zinc finger. In this case, residues 1 to 10 form an antiparallel hairpin motif with cysteine 3 in first beta strand and cysteine 6 in tight turn between first and second beta strand. The hairpin is followed by alpha helix from residues 12 to 24. The helix is distorted between histidine 19 and histidine 25 to form a 310 helix. Structure of ZIF268, a mouse protein with 10 base pairs of DNA. Again, this is the first example where the structure with the DNA molecule of zinc fingers was available. So it is important how the zinc finger bound to DNA. In this case, the 90 residue peptide fragments consist of 3 zinc fingers. The 12 residues between second cysteine and first histidine belonging to beta sheet, loop and alpha helix form the main interaction area with DNA. The interactions are both specific as well as non-specific. ZIF268 DNA binding. In ZIF268, 5 polypeptide chains bind to DNA. Arginine 46 at end of loop has 2 H bonds with guanine in 7th base pair. Histidine 49 forms H bond with guanine of base pair 6. Base pairs 2, 5, 6 phosphate groups interact non-specifically with the side chains of histidine 53, arginine 42 and serine 45. Histidine 53 stabilizes zinc finger through interaction with imidazole, nitrogen, and participate in H bonding. Zinc-containing motif of glucocorticoid receptor. The glucocorticoid receptor belongs to the family of transcription factors that include thyroid hormone receptor, retinoic acid receptor, vitamin D3 receptor, and steroid hormone receptors. Protein fragments produced from recombinant DNA technology have sequence-specific binding to glucocorticoid responsive element, GRE. Structure of glucocorticoid receptor. The amino acid sequencing glucocorticoid receptor shows that each zinc atom is connected to 4 cysteine residues. The 3D structure shows that the 2 zinc binding domains are interwoven into a single globular unit. Second pair of cysteine initiates an amphipathic alpha helix. The hydrophobic core of the 2 alpha helices pack against each other to form a compact core with hydrophobic interior. We show in this picture schematically the amino acid sequence of glucocorticoid receptor. There is a first zinc finger where 4 cysteines bind to zinc, and there is a second zinc finger where again the 4 cysteines bind to zinc atom. Both the fingers are followed by helical region. There is a P box and the D box. Specific DNA binding sequence are in this P and D boxes. Glucocorticoid receptor DNA binding. The 2 zinc atoms shown in green and the protein region in between zinc ligand form protrusions from the globular core. Leucine and others studied the binding of glucocorticoid receptor with 14 base pair DNA from GRE. The structure 1GLU PDBID is shown in the figure on the left hand side. The glucocorticoid DNA binding. The GRE comprises of 2 palindromic half sites separated by 3 base pair spacer region with sequence AGAACA NNN TGTT-CT. The spacer region sequence is unimportant, but it is important for proper binding. Its length is important. The DNA binding domain of receptors are monomers in solution, but dimerize upon binding. This dimerization is crucial for binding of GRE. If the 3 residue spacer is replaced by a 4 residue spacer, the receptor binds only as a monomer, displacing the second monomer. Glucocorticoid receptor DNA binding. It's seen that binding to DNA initiate substantial conformational changes in the dimer interface. Most of the interdomain contacts are made between the first 2 cysteine ligands of the second zinc finger. The specific base interactions are provided by a helix of the first zinc finger motif. The second helix has number of non-specific contacts. Lysine 461, valine 462, and arginine 466 make specific contacts with the edge of the bases in minor group. Leucine Zipper. This is another structural motif which binds to DNA. The sequence of these motifs, when plotted on the helical wheel of alpha helix, shows that leucine residues all of them lie on one side of the field. The peptide dimerases because of the hydrophobic contacts among the leucine side chains to form two parallel coiled coil alpha helical lipids of 3.5 residues each. Basic region leucine zipper B-zip GCN4 is a typical protein in this class. It is divided into two regions. Basic region of about 20 amino acids followed by C-terminal of leucine zipper. The basic region contains each 8 charged residues as arginine is involved in binding. Helix-Loop-helix DNA binding motif. Here, these have substantial amino acid identity and they bind to consensus DNA sequence like CA-NNTG. The myogenic proteins MYOD are important class of BHLH transcription factors. Latter are crucially involved in development of muscle cells. We show here a typical structure where there are two helices and a big loop in between the two, and they bind to DNA. So students, what have we learnt in this today's presentation? First, we understood the basic concepts behind protein-DNA interactions. Next, we tried to understand recognition of DNA-based sequence by prokaryotic transcription factors with helix, turn and helix motifs. We have also gone over non-specific interaction between DNA and histone proteins. We have considered recognition of DNA-based sequence by eukaryotic transcription factors containing zinc fingers. We have described you the DNA binding to leucine zippers, B-zip structures and BHLH structure. These are very important because they have both a zipper leucine-leucine interaction and a basic which holds the DNA. Lastly, we have introduced you to an important topic of binding and recognition of a single-stranded DNA, that is ssDNA, by single-stranded DNA binding protein SSB. I guess that this presentation will let you understand importance between the interaction between the DNA and the protein molecule. Both the molecules have great importance because it is the proteins which control all our function, but it is the DNA which controls synthesis of protein through specific interactions and control the protein synthesis. So, this specific recognition of proteins and DNA plays great important role in the biology and important for designing drugs and controlling disease condition. Thank you.